Over the last few episodes, we've unpacked how to test a drug's cardiac safety. The in vitro single channels, the virtual heart in a computer, the beating stem cell heart muscle. But testing isn't the finish line. All that data must become a report the regulators accept before a drug can actually reach the market. How that report is written and by what rules hides in two cryptic codes, S7B and E14. Miss them, and tens of millions spent on data can be wasted. First, two tracks. A drug's cardiac safety assessment walks on two legs. One is non-clinical, done in animals and cells, with the rules written in ICH S7B, governing whether it can be given to humans. The other is clinical, done in real people, with the rules in E14, governing what actually happens once taken. S7B is the home turf of the patch clamp work we've covered. Today, we focus on E14. At the heart of E14 is a dedicated clinical trial, the Thorough QT study, or TQT. You take a group of healthy volunteers, give them the drug, and repeatedly measure their QT interval with high-precision ECG to see if it stretches. To prove the trial is sensitive enough, you add a positive control, usually moxifloxacin, an antibiotic known to mildly prolong QT. In the end, you watch one number. Whether the drug's QTc change crosses a warning line of about 10 milliseconds. But TQT has a big problem. It's expensive and slow. It's a standalone trial designed just for QT, often costing several $1 million and eating extra development time. Not every drug is worth running one for. So a natural question arises. Can that money and time be saved? In 2022, a shift gave the answer. S7B and E14 issued a joint set of questions and answers that brought the mechanistic data we've described to the front. If your non-clinical evidence is solid enough, a best practice hERG assay plus an in vivo QT study, both negative, the double negative from episode 2, then regulators may let you reduce or even replace that costly dedicated TQT study. This is exactly where patch clamp and CiPA data cash in their value. It can go further still. The new paradigm is concentration QTc analysis, CQTC. Instead of a standalone TQT, it draws a few extra tubes of blood during the earliest clinical trials, the first time the drug is given to humans, recording drug concentration and ECG together, then uses a model to link how much drug is in the blood directly to how much QT changed. Two birds with one stone. An entire dedicated trial saved, with data that's earlier, more continuous, and more convincing. Now you can see a complete chain of evidence. From an IC50 measured by patch clamp, to a QNet computed by the virtual heart, to a field potential recorded from stem cell muscle, to a concentration QTc in the clinic. Each link connected, finally written into a report the regulators accept before a drug's cardiac safety truly clears. This is where that patch clamp in your lab ultimately connects, letting a good drug reach the market and stopping a bad one early. Season 1, Cardiac Safety, closes here as a complete loop. Next time, we step back and reassemble all these channels into one full heartbeat to see exactly who governs each phase of the action potential. Follow Channel Observatory, and we'll take them one channel at a time.